I said, you just have to design the experiment. You do not have to do it. So give the hypothesis, okay, for your experiment. So typically, how you would measure photosynthesis is the growth rate of the plant. So is it going to get taller, wider? Is it going to grow more leaves? You have to have it in like a measurable product, right? You just can't say the plant will get bigger. Well, what does that mean? Okay, it's pretty vague. So, plant will get taller, a plant will grow more leaves, something like that. So, if I put a plant in sunlight and one in darkness, the plant in the sunlight will get taller, something like that. Okay. So, the independent variable is what you are testing. Okay. So, you are testing the rate of the amount of light in this case. Okay. If you were to give one plant fertilizer and the other no fertilizer, you would be testing the effect of fertilizer. Okay. Dependent variable should be the rate of photosynthesis, or how tall the plant is getting. Okay. What are you measuring? Okay. Dependent variable is what you are measuring. Alrighty. So give a full detailed list of materials. Now, every single thing that you use for these plants must be the same, except for one thing. All right. So if you say, like, I'm going to put one in sunlight, one in darkness, give them different amounts of water, different amount, give one fertilizer, one no fertilizer, you're not going to know which one of those variables caused your change. Okay. So you want to have one variable only. So let's say you're choosing, for the sake of better argument, let's say you're choosing fertilizer. Okay, so what you would do is you would give me, you would say I will use the same potting soil, same sized pot, same type of plant, give them equal water, equal amounts of sun, except I'm using fertilizer in one and not giving it to the other. Okay. How are you going to measure? Your unit should be in metric. Okay, if you're measuring like height, right? So say I will measure each plant in centimeters, okay, to see which one grows taller. Alrighty. Measurements, how the measurements will be taken. Are you going to take them every 2 days? Every day. How long are you going to do the experiment for? Okay. Okay. Are you developing a color scale? What are you measuring, and how are you measuring it? Okay. Give me the step-by-step procedure. Okay. Make sure it is thorough. Like if I wanted to go home and do the experiment myself, I should be able to, based on your description. Okay. So I used 2 cups of potting soil for each plant. Each was in a quart pot. I used 2 daffodils, whatever. Okay. Stuck each on a westward facing window, for a week. All right. I added, let's say 1 cup of fertilizer to plant A and didn't give any fertilizer to plant B. Watered them every day, at 4 o'clock, whatever. Okay. So make sure every step is given. Okay. And I took measurements, every other day. At the end of however long your experiment, your hypothetical experiment, where you do not have to complete this. So at the end of 2 weeks or a month, however long it is, I'm going to see my final results, and that's it. Okay, you just have to design it. You don't have to actually complete it. So, like, you can also use the amount of light. You can put one in by a window and one in a dark corner. Make sure everything's the same amount, except for the sunlight. Amount of the soil, the type of soil you use, the amount of water you give them, the time you give them each water. Okay, that sort of thing. Okay, make sure that's the same plant because different plants have different growth rates. All right, don't measure, don't do like pea plants and then a rose. You know what I mean? It's not the same. Okay. Alrighty, so that's lesson 21. Why that's in the genetics one, I don't know. But, okay, we're moving on. So lesson 22, these are all computer graded. They're all multiple choice questions. Okay? So these are all multiple choice questions. Alrighty, so you click here to open the data sheet. Right here. Okay. Okay, so you're going to go through, there are 10 traits. Widow's peak. That's a widow's peak. Do you have one or not? Okay. Can you roll your tongue? Yes or no? Okay, if you can, if you have one, it's, if you have that trait, it's dominant. If you don't, you have the recessive. Do you have visible hair on your knuckles? That is actually a dominant trait. Okay. Attached earlobes, earlobes that go directly into the side of your head. All right. So, like for example, I have attached earlobes. All right, if you can see, but they go directly meet the side of my head. There's no space here. Okay, so those are attached earlobes. Okay, hitchhiker's thumb, recessive. Does it go back? Well, hang on, mine does. All right. All right. Then go to the second page. Dimples. That's one dominant trait. Bent little finger. My fingers are straight. Freckles. Okay, those are dominant. A cleft chin. Otherwise known as chin dimple. That is dominant. And if your second toe is longer than your big toe, that's also dominant. Okay, so make sure you do all 10 traits. So you're just going to tell me, okay. Oh, whoops, hang on a second. I'm sorry. That's for lesson 23, what I just did. My apologies. All right, so for lesson 23, you're going to go through all those 10 and tell me how many dominant and how many recessive traits you have and which ones you have that are dominant, which ones you have that are recessive. But that's lesson 23. Let's do this. Okay, so classmate has a widow's peak. Okay, so classmate has a widow's peak. So that's the very top one. Okay, so it is dominant. Okay, so the phenotype is dominant. Genotype, okay, for the genotype, you can either be homozygous dominant, so that means you have two dominant alleles, or heterozygous, meaning you have one dominant trait, one dominant gene, and one recessive. Either one will get you a widow's peak. You just need one dominant trait to get you the widow's peak. All right. All right, so dominant, you have either two dominant traits or one dominant, one recessive. Okay, a classmate has attached earlobes. Okay, so attached earlobes, that's the second, or not the second trait, that's down here. Attached earlobes, those are recessive. The only way to get a recessive trait is to have, sorry, I don't know why these all changed. The heck, man. That's not what I wanted. Anyway, so the only way to get a recessive trait to show up is you have to have two recessive genes. Okay, if you have one dominant gene and one recessive, you're going to have a dominant trait show up. If you have two recessive genes, that's when the recessive trait occurs. Okay, so attached earlobes being recessive, there's only one possible genotype for that, and that is little e, little e. Okay. So freckles are also dominant. Okay. Where are the freckles? Okay, so freckles are also dominant. So you can have both traits. Remember, you get one set of genes from each parent. Okay, you get one gene from mom, one from dad. So you get either they can both be dominant, or you have one dominant, one recessive. Because the dominant, by its very nature, will mask the recessive trait. That's what dominant traits do. They're dominant over the recessive. Okay. So freckles are dominant, either homozygous dominant or heterozygous. Okay. So, we can think about it in like terms of homogenized milk. Everything's the same. Homo means the same. Hetero means different in terms of like sexuality. Homosexual, heterosexual, right? Homo means the same. Hetero is different, but either homozygous dominant or homozygous or heterozygous. Okay. So there's two types of homozygous: homozygous dominant and homozygous recessive. All right. But dominant, you can have two different genotypes. Genotype literally means type of genes. What do you have? All right. So that's lesson 22. And I believe I already explained lesson 23. I'll go over it again anyway. Okay. Okay. So you're going to keep the data sheet from lesson 22. All right. So how many dominant and how many recessive traits do you have? Okay, I have, 7 dominant, 3 recessive, whatever. Okay, it has to add up to 10. If you only give me 3 traits, like I've had some students do, I have 2 dominant, 1 recessive. Well, you're going to get a 30 out of 30%. Okay. What accurately explains what determines how often a phenotype occurs? A phenotype is a physical trait. Okay, so how often do brown eyes appear in the population? Okay. So how often the allele appears. Now, their allele is simply a version of a gene. Like you have a gene for eye color, 2 forms of that gene. You get a form from mom, 1 form from dad, and that's an allele. Okay. Those are both alleles. So how often does it occur in the population? Typically, dominant traits, dominant phenotypes do occur more often, typically. Okay. Simply because you can have 2 different gene combinations to have a dominant physical trait appear. You can either be homozygous dominant or heterozygous. To have a recessive trait appear, you have to be homozygous recessive only. So typically dominant traits are more prevalent in the population, typically. Not always the case, but that is typical. Alrighty, so question 3, why you can or cannot know the genotype of a person showing a dominant phenotype just by looking at them. Okay, could you tell if a person is heterozygous or heterozygous dominant if they have a dominant trait? And the answer is no. There's no way to tell because there's 2 different genotypes they could have, right? They could, like for example, for freckles, right? They could either have 2 dominant traits or dominant and recessive. There's no way to know just by looking at them. Okay, you can tell with a recessive trait because a recessive trait only takes 1 genotype. There's only 1 possible way to get a recessive trait, like with attached earlobes. We saw that with little e. All right, you can only have 1 genotype. So the answer for the dominant phenotype, no, you can't tell. You can for a recessive because there's only 1 genotype, but you cannot for a dominant phenotype just by looking at them. Okay. So you can't know if a person's dominant because there's 2 different genotypes. You can tell if a person who's recessive recessive phenotype. You can know their genotype because there's only one. All right, so that's question 3. Okay. Is a dominant trait always seen more in the population? Not necessarily. Okay. The vast majority of the time, yes, but not always. All right. For example, having 5 fingers and 5 toes on each hand and foot is actually recessive. Having 6 fingers is dominant, believe it or not. But having 5 fingers is way more common, even though it's recessive. Okay. All right. Okay, so here's a source you can use, explain why dominant isn't always common. Okay, so you can use that website. Alrighty, to explain for question 4 and question 23. Alrighty, and genetic disorders. Oh, this is research based. Okay. All right, so question 1, you can either do cystic fibrosis or polydactyly. Those are your options. Cystic fibrosis is a genetic disorder where your lungs consistently fill up with mucus. Okay, you typically need a respirator. Polydactyly is the 6 fingers. That's what we talked about. Okay, give definition. What's the cause? Symptoms. Treatment available. Is there a specific population or gender that is more susceptible? Okay, for example, in sickle cell anemia, it's more common in African Americans. All right, Tay-Sachs, which we've learned a little bit about, is more prevalent in Jewish people. Okay. Make sure you cite your source. Okay, and then any other disorder you can think of. Okay. Sickle cell, Tay-Sachs, Down syndrome, Klinefelter's, Turner syndrome. Okay, ADHD, at that, I was pressured into doing that from a former student, but that's not proven to be genetic. So I can take that off. It's not proven to be outright. Okay. You do not have to confine yourself to that list. You can use any one you think of. Okay. So these are just examples that you can use. If you want another one, feel free. Okay. And do the same thing. Definition, the cause, symptoms, treatment, gender, population. Okay. MLA. And some of these, there's no treatment available. For, like, example, cystic fibrosis, there is treatment. You can have a respirator. You can have, steroids to keep your lungs clear, that sort of stuff. Cough medicines, that kind of thing. All right. But for something like Down syndrome, there's no treatment for it. All right. Which is the case in actually most genetic disorders. There's nothing you can do. All right. You can manage the symptoms in some, but for most, there's nothing you can do. All right, sickle cell. Okay. All righty, so click here to get this portion. All righty, and that, my dear, you'll print this out. Okay. Sickle Cell Anemia Lab. All right, please complete it. Hang on, wait for it. All righty. All right. Let's go in. Okay. Question number 1 is computer graded. Where does mutation start? All mutation begins in the DNA. That is where your genetics, all your genes are held, is in your DNA. Okay? So any type of mutation will begin in the DNA. Okay? Alrighty. Now, this packet, if you read it, The first sentence said sickle cell anemia, okay, is caused by mutation in the hemoglobin. Alrighty, that is not correct. So do not go off of this packet for question 1. A lot of kids do that, and it's not right. Okay? The reason it's mutated is due to the DNA. All right? The hemoglobin is mutated. That is true. Okay? But the reason is there's a mutation in the DNA that causes the hemoglobin to be funky. Okay? So don't answer hemoglobin. Okay? Nobody chooses these two, generally speaking. It's either they either get it right or they choose in the hemoglobin. Okay? It wasn't until I printed it out and did the lab myself when I was like, why is everyone choosing this? Oh, because it says it. First sentence. Alrighty? Do not do that. Alright? Okay? So, it says list the nitrogen bases as seen in the DNA sections of the following. So you are literally, okay? So yeah, sickle cell anemia caused by mutation in the hemoglobin. Okay, hemoglobin is a protein that transports oxygen. So what happens in sickle cell, the cell is misshapen, and so the hemoglobin does not transport oxygen. All right, well, it does, but not as well as regular shaped cells. Okay, so when you give the DNA section, you are going to literally list for the normal DNA section GGG, CTT, CTT, TTT. Literally, just write it down. Okay? For the sickle cell mutation, the bases GGG, CAA, CTT, TTT. Okay? So that is the answer to part 1. Question 2, what nitrogen bases in the sickle cell mutation section are different? Okay? They are right here. Alrighty? They are base pairs. You can count them off. 1, 2, 3, 4, 5, and 6. And they are both adenine. They should be thymine. That's it. So base pairs 5 and 6 are both adenine. That's the only difference in the DNA. 2 base pairs. That's it. Okay. All right. So every 3 bases. So just like you don't read letter by letter, you read words, right? DNA works the same way, except every word is 3 bases long. Okay? So their words are called codons. Alrighty? So there are 1 codon, 2 codons, 3 codons, 4 codons in the normal DNA section. A codon is 3 bases. So every 3 bases, it's a codon. That's how I came up with 4. So count off by 3s. 1, so go 1, 2, 3, 4. All right? Same thing for the sickle cell. 1, 2, 3, 4. So there's 4 codons in each strand. Okay? All righty. How many codons are on the following? 4 each. Use the amino acid code section. How many amino acids are on the following? Okay? So, alrighty, so using the amino acid section, so what you're going to do is you're going to fill out your sheet. Okay, you're going to go to page 2. Okay, and you're going to fill out the mRNA code, and then the sickle cell for both DNA strands. All right, now for mRNA, alrighty, okay, you're using for this table for your data sheet, you're using the DNA strands on the bottom of page 1. Alrighty, and you're using the pairs. All right, so G and C pair up, T pairs with A. Okay, so, all right, so let's do this. So I'm just going to throw in, okay, let's do it one codon at a time. All right, so G, G, G. Since G pairs with C, okay, the mRNA is going to be the complement, okay, the pair of the DNA strand. All right, so the first 3 letters in the DNA are G, so the mRNA has to be C's. Okay, so CTT. All righty, you can follow these rules here as well. Okay, so C goes to G, T becomes A, GA. All right, and then CTT, C becomes G, T becomes A, so TT becomes AA. And lastly, 3 T's, T turns into A, and that's it. Okay, for the sickle cell, first 3 is GGG, same as the normal strand so far, CCC. All right, because that's what we're using. G turns into C. Okay, you do not have to do the tRNA. You can if you want to, but you don't have to. All right. The tRNA strand should look exactly like the DNA, except for one base pair. Okay, so RNA uses uracil instead of thymine. That's the difference. Okay, and RNA is also single-stranded, but it's still A and U still pair up, same as A and T. Okay, so CAA. All right, so I know that C becomes G. Now A turns into U. So now it's different. Instead of GAA, it's now GUU. Okay. Go back up here. Third set of 3. CTT becomes GAA. And then lastly, TTT, same as the original AA. All right. So there are 4 amino acids coded for each strand. Okay, so the question asks you, using the amino acid code section, term amino acids are on the following 4. So there's 4 codons on each strand and 4 amino acids on each strand. Alrighty. Question 4 asks you to order them. Alrighty, so what is the order? Okay, so you're going to use this table right here. Alright, we're going to skip over the tRNA. You do not have to do that. Okay, so you don't have to do it. Order of amino acids. Alright, so CCC gives you amino acid A. GAA gives you amino acid B. Oh, GAA again also gives you amino acid B. AAA gives you amino acid C. Okay. Sickle cell, CCC gives you amino acid A. Then we have GUU. This is where it's different. GUU gives you amino acid S. Okay, GAA gives you amino acid B. And then AAA gives you amino acid C. Okay. Boop. Alrighty. The shape here is round. Shape here is sickle. So the only difference in normal brown blood cells that are, elastic, like they're like an inner tube, right? They can squeeze into capillaries, they can go places. There's that big divot where they can hold lots of oxygen. Okay. The only difference between that and sickle cell, which are like crescent shaped and don't carry much oxygen, and that the points get caught in your capillaries and cause painful. The only difference is one amino acid. That's it. 2 base pairs, and boom, you have sickle cell anemia. Okay, that's it. Okay. So question 5, how sickle cell mutation affects red blood cells? Well, it changes their shape, makes them crescent shaped, like a crescent moon, or like the Pillsbury crescent rolls, like in the letter C. Alrighty. So those points will get caught. They don't flow through blood vessels well. So they get caught and you get blood clots and it becomes painful. Okay? The points get caught on the edges of veins, capillaries, and it becomes painful. You get blood clots. Also, because of their weird shape, they don't carry much hemoglobin. So because they don't carry much hemoglobin, okay, because they don't carry much hemoglobin, they don't carry much oxygen. And that's what being anemic is that your blood isn't carrying enough oxygen to where it should be. You get tired and run down. Alrighty. Whereas round cells, they can go through, they can squeeze, they can expand. Alrighty. They got the big divot where they can carry lots of oxygen. Sickle cells can't do that because their shape is weird. They get caught on the sides of your blood vessels, causing clots, and they don't carry much oxygen. So that's the answer to 5. Okay. Alrighty. So as you can see, alrighty, I did this all myself. I even did the tRNA. Okay. Except the difference in here with the tRNA, okay, the difference is that the T's for the original DNA strand, which I'll show you. Whoops, hang on a second. See how it's all T's at the end? There's a couple T's here as well. Alrighty. Everything is U's. That's the difference. So it should all be the same, okay, on the other side as well. The tRNA is the same as the original DNA strand, except change the T's to U's. That's it. All right? Because RNA uses uracil, not thymine. Okay, and they're single-stranded, not double-stranded like DNA is. Okay? Cool in the gang? Alrighty, Jacqueline. Oh, stop recording.